it's very important that for our orchard management we try to minimize the input and maximize the output in our system to obtain the maximum efficiency and an economic and ecological sustainability. For example, we can do that with precision horticulture methods. For instance, water saving in orchard management is very important, especially here in Australia. It's becoming more and more important in the last few years. But to do that, to save water efficiently, we need to know when and how much water to provide to our trees. And to do that, we can assess soil water status, but it would be much better to assess plant water status because we would answer the real plant needs. So are there any methods for the continuous determination of tree water status? In this study, we had some few hypotheses. Two of them were that continuous fruit growth rates and leaf turgor pressure dynamics change under different irrigations in nectarines, and the deficit irrigation applied at different fruit growth stages can affect tree physiology in different ways. And the objectives were to find out the most sensitive continuous indicators of water deficit in nectarines and test different portable non-destructive devices for determination in field of leaf and fruit composition. And the specific research questions were, are fruit growth and leaf turgor pressure related to each other, and can we associate them to the reference method of tree water status assessment, which is stem water potential? And then, are near-infrared and fluorescence spectroscopy suitable for non-destructive determination in the field of leaf composition and fruit composition? For example, sugars, dry matter, and flavonoids. So we worked in an open Tatura system with a drip irrigation system, and we had 144 measurement trees at a planting density of 2,200 trees per hectare. And the trees were 4 years old, and we worked with one genotype, September Bride, nectarines, grafted on Alberta rootstock. We had 6 randomized blocks in a randomized block design with I can't see what's up there. Yeah, 12 irrigation treatments, 2 canopy orientation treatments for a total of 144 measurement trees. So we had 12 irrigation treatments, as you can see here. Where's this thing on? How can I? 12 irrigation treatments with 0%, 20%, 40%, and full irrigation of 100% of evapotranspiration, crop evapotranspiration. And we had these 4 irrigation levels within 4 different stages of fruit growth. And the stages of fruit growth, 1, 2, 3A, and 3B, were chosen according to the different cell division, P-target, and cell expansion stages. And then the stage 3 was subdivided in 2 according to the different moments in which actually there is an increase of sugar accumulation and chlorophyll degradation. And then we had 2 different canopy orientation treatments, west and east trees. Then we also used the time as a factor in our statistical analysis. This is the list of all the equipment that we used in our experiment. So it's pretty plenty of different devices. The 2 continuous sensors are the fruit gauge and the zinc probe, or LPCP probes, which means leaf patch plant pressure probes. And they measure continuously fruit size, fruit growth dynamics, and leaf turgor pressure dynamics. Oh, sorry. We've got many results, but I don't have enough time to present all of them, so I've chosen some that I think are more relevant for my work here. And the ones I'm going to show you today are the ones in red. This is the overview of the weather and irrigation along the season. So basically we had the most significant things to notice are the big flooding event, big rainfall event in the stage 2, It means like in December we had like almost 14 millimeters of rain in one day, and also the highest temperatures occurred during the stage 3A. And then we had a total of, you can see rainfall plus full irrigation 219 in stage 2, where we had 141 millimeters of rain. And then also the solar radiation was higher in stage 3A. So for what concerns the fluid diameter growth, we can see that aggregating the deficit irrigation in the different stages, we can actually see differences in fluid growth, like the control and the deficit irrigation in stage 2 don't differ much, but deficit irrigation in stage 1 and 3A yielded fluid with a smaller size, and also deficit irrigation in stage 3B was by far the one that had the smallest fluid. We repeated measurements in each of these stages at weekly intervals, and we can see that in stage 1, for example, we do affect fluid size very much with different irrigation strategies, and it's like a gradient from control to 40%, 20%, and then 0%, where we have the smallest fluid. Then stage 2, which is the pit hardening stage, is very well known because we can actually withdraw, like hold the water. We don't need, we can reduce sensibly the irrigation fluid because we're not going to affect fluid growth because it's in the pit hardening stage, and the plant actually is going to reduce the vegetative growth. And stage 3A, we also have a gradient like stage 1, it's a bit different, but we do have a bigger fluids for control treatments. And in stage 3B, we have an even bigger differentiation of size. For what concerns the light trolley measurements and septometry measurements, we calculated the effective area of shade, where we can see that there is an increase in the canopy light interception, except for the deficit irrigation in stage 2, which, as I said, influence vegetative growth. But then after the summer pruning, we have a reduction, of course, of the vegetative area of the canopy, vegetative volume, and then there is an increase towards the end of the season once again. For what concerns diurnal stomatal conductance and photosynthesis, which I don't show because they actually behave in the same way. So there is an expected peak in the morning, like in mid-morning around 10 a.m. And there is a tiny difference in among treatments because we are in stage 2, and water relations are not really influenced by irrigation treatments in stage 2, but there is of course an effect on the canopy orientation, especially on sunny days, because on the west canopy in the morning we have full sunlight, and we have of course higher stomatal conductance compared to the east, and goes the other way around towards the end of the day. But of course this is the part of the day in which we have the highest peak, and of course those trees are going to have higher photosynthetic activity and higher stomatal conductance. The same thing doesn't happen when we have cloudy days, because there are no differences between west and east. But in stage 3A, even in a cloudy day, we have a big differentiation of the stomatal conductance and photosynthesis among the irrigation treatments. Then we use the vigour to measure fluorescence in leaves, and I want to show you the data we've got about the efficiency of the photosystem too, which in the stage 1 is not significant. It's not significantly different among the treatments, while it starts to be different in the stage 3A, where the control is sensibly higher than the deficit irrigation at 0%. And then in the stage 3B, we get the maximum difference, where control efficiency of photosystem 2 is almost double, is more than double than the efficiency in the treatments with 20 and 0% of water applied. The daily stem water potential curves were done in all the stages, but I'm only showing stage 2 and 3A just to show a big difference, where in stage 2, as I said before, water relations are not very much affected by irrigation reduction, and while in stage 3A we still have a gradient where the control have a general overall higher stem water potential compared to the other treatments. And of course the zero irrigation is like the one with the lowest stem water potential. And the reference method for determining like the tree water status is the value of water potential on midday or 1 p.m. when it's the lowest, when we get the lowest values. Then we also use the spud meter to determine the chlorophyll in leaves, and there are not very big differences among the treatments, the irrigation treatments on each stage, but there is a very interesting difference between east and west canopy, where the sun-exposed leaves on the west side get a lower chlorophyll content compared to the east-exposed one, and there is a general trend of increasing chlorophyll until the end of the stage 2, and then there is a big decrease, which probably is due to a conversion of the pigments into anthocyanins and flavonoids and probably other secondary metabolites. So we also use the fluorometer, this fluorometer which has been used only on grapes before. So this is the first time somebody used it actually on stone fruits. And it's interesting because you can determine several indexes which are obtained by ratios between the fluorescence emissions signals. And one of them, which I found interesting, is the flavonol index, which increased from stage 1 to stage 2, to the end of stage 2, and then stay stable until the end of the season. And the difference are not very big anyhow. So irrigation doesn't seem to influence flavonol in leaves, but there is an influence on the canopy orientation, with the east side being with higher flavonols. It means that when leaves are fully exposed to sun, because in the morning, this is the side of the canopy which get fully exposed in the morning, leaves tend to send probably flavonols to the fruit, which I will show you in the next slides. And similar thing happens to anthocyanins in the west and east side of the canopies, whereas there is like an increase in anthocyanins levels in leaves along the season from stage 1 to harvest. About fruit, as I said, there is an inversion both in flavonols and anthocyanins levels in the west side of the canopy, and the irrigation treatments seem to affect the flavonol content and flavonol index in fruits, but it's like a very, very quick and sudden response. So when we withdraw at the beginning of stage 3B, when we withdraw water in the deficit irrigated treatments, we have an increase, or a rapid increase of flavonols, which is then reestablished to the normal levels. It means that probably if we shift this withdrawal of water in the last few days before harvest, we can actually collect fruits, harvest fruit with a higher level of flavonols, which is interesting. And also in the last stage of fruit growth, stage 3B, from 5 February to 4 March, we have a decrease in a general increase of anthocyanins in the fruits, whereas you remember it was the other way around in leaves. And there are not very big differences among irrigation treatments, although there is this pattern of decrease is not confirmed in the deficit irrigated ones in the 3B stage, which actually remains stable to the end. Then we also use this device for near-infrared spectroscopy, and we made a couple of models for dry matter and solid concentration, and we can see that there is an influence of the treatments on the sugars and dry matter, where the deficit irrigation in the stage 3B and 1 yielded like higher dry matter in fluids and solid sugars in fruits. Yes, compared to the irrigation treatments, in the stage 2 and 3A, and the control, which don't look to be significantly different among each other. There is also an effect of canopy orientation on dry matter, where the fruit, which were fully exposed in the morning, so the west side, the west-oriented trees, have a higher dry matter content, probably because they get higher temperatures and they lose a bit of water. And then finally, I want to talk about the continuous sensor that I had on my trees. So we had these fruit gauges and LPCP probes, and what I did is getting the signal outputs from both of them, and I calculated the fruit diameter z-scores and PP z-score. PP is the inverted value of leaf turgor pressure, and this is the output of these probes. And why did I calculate z-scores? Because this is the only way we can compare two different measurement units. And also, among the fruit diameters, I needed to get a zero level for all of them, like for all the fruits with different diameters, so that's why I calculated z-score. And then I calculated the second derivatives of these values, which are the relative growth rate for fruits, and something that I call RPCR, which is the leaf relative pressure change rate in the changing pressure over time. And I tried to see how a regression of them would look like. Very heavy. Yeah. First, I just plot the fruit diameter over time in stage 1, and also the PPZ score. And you can see there are some kind of, it looks like there is some response of treatments. And also I plotted the RGR and RPCR, and I thought like there could be some kind of relationship between the two of them and these two of them. So what I've done was doing a regression between fruit diameter and PP and RGR and RPCR. What's wrong? Okay, so I've done a regression in stage 1 among RPCR, between RPCR and RGR, and also fruit diameter and PP. And this is really like a cloud of points. You don't really understand much of it. But this is like typical because I've already studied this regression on olive, and I've seen this behavior, the same behavior happening in olive. And this is probably due to something that I didn't have time to plot it for nectarines, but this is happening in olives. So there is kind of a hysteresis during night and day. So the relationships are different between night and day. And that's why you see all this amplitude of fluid diameter points along the PP range. And same thing happens in stage 3B, where you see all these weird amplitude things. So what I did was trying to have a ratio between the values of PP and fruit diameter, and also RGR and RPCR, and try to regress this index to the reference method of free water status determination, which is midday stem water potential. So I tried to do that, midday stem water potential here, and the way I related the two indexes in the DL values, diurnal and nocturnal, was using the variance overnight, but the only way to compare two different variances with two different amplitudes, orders of data in units, because this is, we remember, this is millimeter and this is kilopascal, was calculating the coefficients of variation. So I actually made a ratio between the two coefficients of variation, and then I plot the regression against midday stem water potential. And they look strange, they don't look so nice. And also, if you think this might look nice, then you see on the stage 3B, it looks like completely different compared to this. So what I did was merging the two stages, and I had something that looked much better, especially because I did the same, like DL, diurnal, and nocturnal for all the ratios plotted against stem water potential. And the only thing I found interesting was what happens at night. So when I have this ratio between the two coefficients of variation of RGR and RPCR plotted against midday stem water potential, I do have this kind of relationship, which didn't look strange to me because it's exactly what I found in olive. Exactly what I found in olive. And the nice thing in olive, which I still didn't have time to, verify here, is that I was working with two very different genotypes, like very different in the way to react to water deficit. And I found in the t-test between the two slopes, I found no differences. It means that there is something like in the leaf to fruit balance and water movements that looks to be regular among, for sure, genotypes in the same species, and then trying to see if it works also among different species. So that's, of course, that would be an amazing thing if it is true. And our conclusions are that the combined index of fruit growth and leaf turgor pressure is more powerful compared to considering them individually. And this integrated approach for continuous monitoring of water status in plant organs can be used, of course, for automated irrigation management. And if we set some threshold of stem water potential, midday stem water potential, we can definitely, using an Arduino board or any kind of board, we can definitely manipulate. I mean, we can just automate our irrigation system and not think about it anymore, especially if we have a relationship that works for all the fruit growth stages. That would be really interesting. And also the fruit quality parameters may be determined by non-destructive field measurements, by near spectroscopy and fluorescence spectroscopy, and that's what actually some people is doing in Europe. So they're trying to develop some sensors, some fluorescence sensors, which can be kept continuously on the trees. And yeah, these technologies might potentially be automated and have regular real-time determination of desired parameters. So yeah, what are the problems that I especially found with the zinc probes, with the small leaf probes? So you get some small damage on leaves, you see some kind of bruise. That's pretty common. Sometimes you find leaves that are completely off, and then of course you can find some redback spiders. You're censored. So thank you very much for all the people that have been helping me during this period, and it has been great. I can't believe I'm leaving. Yeah, and especially I want to thank also my family in Italy, because sometimes we just forget how important our parents follow what we do, but we should never forget. Thank you. And this is a paper a review we published a few months ago. If you want to read it, I'd be really happy of that. Thank you.